Okay, good afternoon everyone. Welcome to the session on predicting non-effect concentrations of antimicrobials. It's an honor and pleasure to share this session together with Thibaut Stalter. We will be sharing this session together, and the first speaker will be William Gaze. Will has been working on AMR for more than 20 years, now together with Aimee Murray and Leonard, who can't be here, and he has increasingly been interested in relating his research into policy and has made some important contributions to reports like the United Nations Environment Programme and EFSA, World Health Organization, and others, but I don't.
Right, thank you very much. So I hope I can get all these buttons right. So thank you very much for the invitation. I'm going to talk about, I said it's a very vague title, but predicting, investigating AMR evolution in the environment. And then I said in the pursuit of PNECs, which I suppose is because actually I think that PNECs are just one thing that's important, and it's really not the only thing by any means. So I'm at the University of Exeter, but actually I'm right down here in Cornwall at the end here near Falmouth, and that's the campus, and that's Falmouth Harbour. It's the third deepest natural harbour in the world, and actually where the Mayflower left for the US, and I think where the Beagle with Darwin returned. So because it's the biggest port on the furthest southwest. So these are the people that contributed to what I'm going to talk to you about. Some currently, some more historically. As Jan said, that Aimee has done a lot of the work on this. Anyway, so these are the other people. So this work is all funded by UK Research and Innovation, with a small amount of funding from AstraZeneca in top-ups to studentships for, I think, 3 of our group. So I expect you've seen this before. I missed Jerry's talk. But I decided now I start every single talk with this slide, because as we discussed yesterday, with these high-level policy communications about AMR, this whole origin story it never seems to make it into things like the national action plans and other things. I mean, of course, it's in the UK, of course, it's in the UNEP report, but in the really highest level things, and for example, as we heard in the UN General Assembly. It's not going to be mentioned. So also, again, another of Jerry Wright's figures, this one about the ancient permafrost, but this is really just to emphasize that, this has been mentioned before today, but, there's this acute AMR pathogen transmission risk, and actually I think 99% of people working on AMR, they're really only talking about this. But most of us are talking about this process, this sort of like epidemic, almost like pandemic emergence, which parallels with emerging zoonoses in many ways. So there are all these different diagrams of like One Health systems and system maps and things, and this one is the basis for the Canadian, I think 2000 confusogram on AMR. But actually, I think this is actually the most useful one in many ways, and this is from 1977. So, One Health, it's not a new thing. They didn't call it One Health then necessarily, or maybe that was I can't remember when the actual terminology emerged for the connection between animals and humans. But so I have made a modification to this, and I've just put this arrow here, this is selection. So we've got high selection in humans, lower selection in the community, so hospitalized rather, lower in the community, high selection in animals, and I've just copied and put this low selection in the environment. But really, this is just as current. So when we're thinking about PNECs, I suppose, in terms of the environment, we're really asking the question, is like, how strong is this selection? But I think the critical thing is about PNECs themselves. They just tell you what the threshold where selection might occur, but they don't tell you anything about the nature of that selection in the microbiomes above that threshold. So I think it's one of several methods that we need to really understand what's going on in terms of evolution and emergence. And Joachim mentioned in his talk as well that there are different scenarios, aren't there? So there's potentially that we might have human or animal-associated bacteria going into the environment under selection where new genes are acquired, but maybe that all happens in the human. This laser pointer seems to appear and then disappear. Maybe all the important transfer events happen in the human microbiome, but actually we don't really know. So some of what I'm going to say to you is that actually we need those experimental approaches to try and understand that, as well as being able to define these selective concentrations. So going back from this 20 years, and this is the work that I did, we did in Liz Wellington's lab, so I was a part of her group for 12 years. And this is work that Greg actually did, and I didn't put him on the front because I couldn't fit him on. But and this was work done with Andrew Singer on the River Thames. So we were looking basically at levels of INTI1 prevalence within different sediments, four samples over about a year. And then we basically looked at the distance of the sample site from all the sewage treatment works within a 10 or 20 kilometre radius. And then we did some simple sort of statistical modelling, and basically we could predict the levels of resistance as indicated by the INTI1 prevalence with quite high certainty, and that was basically based on distance from type of and size of wastewater treatment plant. So Greg also did some work looking at upstream and downstream, a really large wastewater treatment plant discharging quite a small river, and I think was the first to identify CTXM bearing bacteria or E. coli in aquatic sediments. But this paper actually I think was really interesting and we haven't really even followed up on the significance of it. So we found CTXM15 in multiple different species, including Aeromonas, which we might think is an aquatic organism, but is another one of these opportunist pathogens. And when Greg then sequenced out using Sanger sequencing in those days, because this was 2009 and 2011, I think the sample time points were, that we found that there was like 10 novel genetic contexts of CTXM15s, which indicated that it was translocating, frequently. And in some cases, these isolates were resistant to over 1,000 microgram per mil, so incredibly high levels. So we could just be looking at diversity from the human gut, but actually it's very difficult to know because that level of probably scrutiny has not been put on CTXM-bearing organisms necessarily. But it was sort of big, we began to think, well, maybe selection is occurring in these sediments. And then, of course, this paper came out, which you're probably aware of, the Goldberg paper from Dan Anderson's group. And interestingly, this came out in 2011. And in 2012, we were in EDAR7, as we now call it, in Montebello. And I think Joachim and I and others, we were discussing this paper. And I just moved to Exeter University, and I was working with all these evolutionary biologists. And this seemed really an interesting approach. Rather than just trying to infer things from descriptive fieldwork, we could actually test whether resistance was occurring in representative microbiomes. So just to take a step back then, so in terms of PNECs, so historically, the predicted no effect concentration, of course, is of compounds or drugs is determined in ecotoxicological assays, which are very simple. They're not environmentally realistic, but that's what ecotoxicology does, and we accept the output of that, and we use that to inform risk assessment. So these are the drugs that were on the original Water Framework Directive, EU Water Framework Directive hazardous compound watch list. So these are compounds that have been sort of marked out as potentially being important from an environmental perspective, and these are tested against these single organisms. So Daphnia or cyanobacteria, and these are the concentrations which were determined, and these are way below the concentrations that are measured in the environment. So that's why they were placed on the watch list. And then since then, there have been different iterations of the watch list, and in this iteration, which I think is the third iteration, the Joint Research Centre in the European Union started to consider this idea of selective potential of antibiotics. And and also in this situation as well, it's also mentioned. So and that work was, there's many people that have been working in this area, but myself and Joachim's group are also referenced in these documents. So there's been different methods that have been used to try and approach this problem or question of what the selective concentrations of antibiotics are. You can see that some of these slides are nicer than others. That's because they're Aimee Murray's slides, and I've nicked them and put them into my presentation. So in 2003, there was this Klaus Kummerer who's been incredibly prolific and worked in this area. You know, they used, well, I'll talk about a bit more detail in a minute. But then anyway, so there are these MIC-based approaches which we know and have been incredibly impactful in informing the industry alliance thresholds, and that Johan Bengtsson-Palm and Joachim have been working really hard to inform, you know, policy in this area. Then there are these sort of competition assays, and this is probably, maybe this is the start, or there was a few studies before this. But really, that's the Goldberg paper. That was with these single-species competition assays. And then there are these multi-species ones. So as usual, Joachim got there first and got the paper out first. But I think we started to work on this thing probably roughly at the same time, just that we're always much slower. So anyway, so there's these bodies of evidence coming from multiple different approaches. And actually what I think is that they're not really in competition with each other. They actually all tell us quite different things. So this is the Kummerer paper with the MIC 50s, and those values were above measured environmental concentration, so he concluded that there was a probability that selection was happening in the environment. And then there's that paper I was mentioning with Johan and Joachim, and I'll probably get this wrong, but this was using the lowest 1% of the MICs in the UCAS database, and then applying an assessment factor, and that's then how you derive it. So essentially, I've done the wrong thing. So essentially, the PNECs are derived from the MICs of the very most susceptible human pathogens, and that, you know, that produced a comprehensive set of data which then that can be used to inform risk assessment. And then there's the single species competition assays, and this is from the Goldberg paper. And I don't know if you've all seen this before. I think maybe someone has already shown it. Maybe that was in the workshop. But essentially, it's the sub-MIC selection window where basically you find that resistant bacteria, although it's below the inhibitory concentrations, the resistant strains are growing faster than the susceptible strains. But where these lines cross, these growth rate lines cross, is defined as the minimal selective concentration. And that's where basically the resistant and the susceptible strains remain balanced in terms of their ratios. And in this study, it was done using GFP and RFP labeled strains and flow cytometry, so very accurate. You could see shifts in the ratio when you grow these organisms across a whole range of different concentrations. And then this is the sort of data that they produce. So basically, you look at the ratio of resistant to susceptible, and each of these is at the different concentration over time. And then they are translated into a selection coefficient and plotted. And where this curve crosses the x-axis, that's the minimal selective concentration. But even when you start to look at this, you can start to see that, you know, this is the sort of the fitness cost in the absence of selection, which is on the, this keeps disappearing, which is here at the bottom, on the bottom of the y-axis. And you can see if you look across these are different compounds. So it's streptomycin, then you've got ciprofloxacin, top right, and tetracycline and ciprofloxacin. You can see there's a relationship between the fitness cost and the minimal selective concentration. And you can also see that the difference between the MIC and the MSC can be very small. So at the top left here, we've got the MSC being a quarter of the MIC. And then bottom right, you've got a fluoroquinolone resistant mutation, actually, and that's over 1/200th of the minimal selective of the MIC. And then we go on to the complex community experiments, which essentially is doing a similar thing, but inoculating microcosms with whatever you want to use. In this case, most of our work's been done using wastewater communities, so dominated by human organisms. And arguably, that's what we're interested in, I suppose, in terms of where resistance might emerge. And then you can use qPCR, or you can use plating, or you can use metagenomics to look at multiple endpoints and try and define these selective thresholds. So this is what Izzy over here did for her PhD on before she went to UKCH. So this is the type of data, and this is Izzy's data. So what we find is that this is the concentration of ciprofloxacin, and this is using INTI-1 qPCR just as a marker, and that you find somewhere you see this sort of biological effect where you see the prevalence increases. These are quite noisy by definition. We do like multiple replicates, about 5 replicates, but you find there are founder effects in the different replicates because they're only pseudo replicates really if you're inoculating with wastewater. And this is the minimal selective concentration figure for the same compound. But what we noticed was that some of these minimal selective concentration curves start to look a bit strange, and actually maybe they're a function of the concentration range that you use. And also we started to see that for some compounds there is no fitness cost, so basically this line never crosses the x-axis, so you can't define an MSC. So we started sort of using this lowest observed effect concentration, and then which then in turn gives you a no effect concentration, which is the concentration below. Then you use the assessment factor, and then you generate your PNEC from that. So from this, we would have a PNEC of 0.78 derived from the LOIC or the NOEC, and I think 1.077 from the MSC. And then you can see for the ecotox PNEC, it's about 0.1 or just below that, and then for the MIC-based PNEC, by 0.064. So these complex community ones are higher, but for ciprofloxacin, they align relatively well, but for some other compounds, they align far less well. And so there are still outstanding questions about why that is. So is that something to do with the methods? It could be that it's something to do with this complex community experiments, or it could be something to do with the fact that those very highly susceptible pathogens that we've got MICs for are not represented in these communities or actually in any environmental community. But again, this is something that we need to discover. Also, when we were doing this work, when Izzy was, the royal we, isn't it? So when Izzy was doing this work, we realised that actually that there's something else going on. And Joachim and I have had long conversations over email about this. And you can even see here that, so these concentrations here that are decreasing, so this is the absence of antibiotic, this is the next highest concentration, So there is something going on in this sub-MSC concentration. The fitness cost close to the MSC is much less than it is in the absence of antibiotic. So this is actually, the definition of the MSC is where you get enrichment, so it's positive selection, but actually in this sub-MSC window, you get differential rates of negative selection or persistence. So, for example, close to the MSC, the minimal selective concentration, but actually below it, you might be seeing persistence lasting for longer. Actually, I've just remembered we've got the figure, and that is the figure I'm going to show you in a second. So, but this is not that figure, but this is another different figure. So we've adapted the Goldberg one, which is actually, you can see that in the original Goldberg figure. Actually, this is there, but what we did is just extended this really, and this shows that basically that there's this window where you see differential persistence of resistance below the minimal selective concentration. And to try and explain this in a simpler way, this is a figure from a report that Exeter and UKCH, so with Holly and Izzy and Andrew and Aimee and myself, but I can't talk about that unfortunately for various reasons. such is life. But anyway, so this is basically positive selection above the minimal selective concentration. You get enrichment. This is at the MSC, so you get this balance between resistant and susceptible. And then this is the sub-MSC selection. So this is this increased persistence. And then this curve here, right at the bottom, that's in total absence. So you can see that actually these sub-MSC concentrations are still contributing to prevalence in the environment, even below that concentration which we might define as being the threshold. So this is quite a complicated thing, and so you know, what do you use for your threshold, you know, or your level? Do you use the minimal selective concentration or the lowest observed effect concentration for enrichment, or do you use a lower concentration where you see this increased persistence? Right, okay, so I'm just going to show you now a couple of the papers of Aimee's papers to show you what these experiments can generate that, you know, the sort of estimated PNEC methods don't. But of course, these are slow. They also have biases, and they're more expensive. So this is for cefotaxime. So basically, this is two different experiments because we couldn't run, or Aimee couldn't run all of the replicates in one go. So what we see is across 5 orders of magnitude of cefotaxime concentration, we see an increase in, or selection for, this is CTXMs actually, CTXM genes in the complex community. And then we get this plateau. So there isn't a linear relationship between selective pressure and selection. So this is, again, one of these, we might think that's logical, but you need this type of experiment really to tell you whether that's true or not. And the other interesting thing was we saw that the CTXM-bearing organisms were outcompeting all the other ESBL-bearing organisms. You know, and, you know, so CTXM have become the global dominant, pandemic extended spectrum beta-lactamase, and lo and behold, even in a little test tube in the lab, we see those organisms outcompeting all the other ESBL-bearing organisms. So, and this is the fold change in these BLARTEM, OXA, and CTXM. So all concentrations, even down here, which is sort of in what you might see in sewers or something, you're getting CTXMs that are out competing everything else, and that are being selected for at the same magnitude or amplitude as at these really high concentrations. So these concentrations go from 2 mg per, so that's the MIC for susceptible E. coli. Of course, MICs don't really mean so much when you're using complex communities. And then we looked at metagenome data, and you find there's this massive co-selection for all different classes of resistance gene as well. And also we could see that some gram-negative and gram-positive pathogens decreased with concentration, and some gram-negative and gram-positives increased with concentration. So you can actually get insights into community structure with these experiments as well. So what have I got? About 8 minutes left. So what we also did, Aimee basically grew susceptible and resistant strains, took the supernatant from the resistant strain and added it with antibiotics to the susceptible strain. And what we find, of course, is that the beta-lactamases, the enzymes that break down the beta-lactams, are excreted and are protecting the susceptible cells within that population. So there's some sort of sociality in terms of resistance. So this is just showing the degradation of, this is the degradation rate of cefotaxime in the presence of the community and without. So basically, it's completely degraded within 9 hours, but we passage into fresh medium every day. So Joachim has pointed out that this might mean that we're underestimating the MSC, which is a good point, because actually it's only at the very beginning of the experiment where you have the full exposure. But I guess maybe that could be happening in the environment as well. And then this is just what I said before. This is where we use the supernatant. So I think if I get this the right way around, so this is the susceptible strain in blue in the presence of 2 microgram per mil, and then this is the susceptible strain in red in the presence of the same level of antibiotic with the supernatant containing the beta-lactamases. And then this is actually Uli's work, Uli worked in Exeter in our group with our colleague, myself and my colleague Angus Buckling for, I think, 4 years, was it, Uli? And he was looking at the effect of community on selection. So do bacteria behave differently when they're embedded in the community than when they're just in isolation growing on an agar plate? And I'm sorry running out of time, so to cut a long story short, what he found was that the MSC shifted when in the community, so it shifted up, so you needed more antibiotic to reduce the growth rate. And this is just showing differential growth rates of the organisms in the presence and absence of the community. So, of course, you see lower growth rate of your focal species, in this case, gentamicin-resistant E. coli, I think. So you start to get some idea about sort of what's going on ecologically as well with these experiments. And this is really preliminary, that some work that Li Hongzhang has been doing. And this is basically looking at ciprofloxacin concentration. And this is in IS-20 is high nutrient at 20 degrees, IS-37 is high nutrient at 37, RA is R2A at 20 degrees and at 37. And you can see for those four different conditions, selection happens at roughly the same threshold, but what you will remark on is that this is prevalence of INTI1, which we use as a marker, which it varies between almost, you can hardly see it on this axis, so less than a fraction of 1%, up to 10% or 20%. So if you think that INTI1 prevalence in raw sewage is like 1% or 2%, this is massive. So the threshold is the same, but the amplitude or the magnitude of selection is completely different under different environmental conditions. So you can start to see how different parameters impact these variables. And just quickly, April was doing this work, and Aimee. So you can take some of these PNEC values, you can apply them to measured environmental concentrations. This was for concentrations of antibiotics for ciprofloxacin taken in the UK from all different wastewater treatment plants, and then you can work out this, risk quotient, which is a product of the measured environmental concentration and the predicted no effect concentration, and you can start to predict, well, what's the risk of selection in sewage influent, effluent, and surface waters. Obviously, this risk is based on the PNEC, so if we applied Joachim's PNEC, we'd see greater risk because it's tenfold lower roughly. But anyway, so this is sort of the end point. This is what we want to do for everything what is being done. And then this is April and Laura looked at the data for different areas of the country in influent, effluent, surface water, and you see quite different. You see different patterns for different localities. So you can start to really drill down into the sort of risk and use PNEC to inform that type of information which can be used for decision making. Emily asked me to mention the fact that we've also been looking into doing this in fungal communities, but now I need to whip on. We've written this report for the Environment Agency, and that's a review of PNEC methods. We've got a database of all PNEC methods in the literature. We're comparing the PNECs which are generated using different methods, and we conclude that MIC-based approaches are generally more protective, but not always, surprisingly. And there's a summary of the pros and cons, so that will be coming out in the next few months, hopefully. So summary, so I think MIC-based PNECs are incredibly useful, and they give you the threshold of selection, and generally they are more protective at the moment than the experimental ones. Some of the single species ones, mind you, were for E. coli only, for ciprofloxacin were very low. So the single species allows you to look at sort of genetic context, whether the resistance mechanism was on a mobile genetic element and how that affects PNEC. And then the community stuff, it gives you ability to start doing more sophisticated experiments. You can look at co-selection, you can look at mixture effects, you can look at changes in community and resistome, you can manipulate the conditions to represent different microbiomes, and you can investigate questions on where evolution of resistant pathogens might have occurred. So you can test hypotheses with it on AMR evolution. And this is the last slide. So I think that all methods, they have their limitations and biases, but they're all capable of contributing to this debate and this discussion. And still, why aren't we doing this in situ? So some people are beginning to do that. Andrew had a grant on it. I think there's something coming, hopefully soon, on the flume experiments where actually antibiotics were added in to flumes actually in a river system. And other people have been doing this sort of thing too. But it's still hugely challenging and really expensive. Thank you.
Might not be a brief question.
No.
Thanks, Will, Joachim here. I appreciate your presentation. I think it was a very nice presentation of the different challenges that we have here. I just want to highlight that you bring up this thing about MSC may not be protective, and the concept that you have introduced with this selective concentration for persistence might be more relevant. And I just wanted to add that there's another reason for that on top of the reason that you have presented here, that it might, sorry, my voice is disappearing, that it's prolonging the lifespan of bacteria. So basically in the nice graph you showed, it's the point where you start reducing growth of the wild type strain. It's that concentration, right? So if we think about it, if we don't know the cost of carrying resistance, and that we assume that it's actually zero, because some of these resistance factors cost extremely little. That is also the concentration that would actually select for resistance in such a situation, where the resistance cost is zero. So and then it would be really a selective concentration that would select for positive selection as well. So I think there are two reasons to consider this point, and that changes the thinking as well, and pushes actually the level of concern even further down, as you point out.
Yeah, no, sure. I mean, with these resistances that have essentially no cost, that's right. They're just going to persist indefinitely.
Yep. And I might ask a question.
Yeah, sure.
You mentioned that it's quite stochastic, the results when you do the replicates, and I was wondering whether the more diverse the communities you're using in your samples, you should be increasing the volume to be able to capture the whole diversity in the sample. If you have thousands of different species and thousands of different genes in even more combinations, the volume you need to be representative would be increasing, which is why some people used to use mesocosms with 3 cubic meters.
Yeah, no,
I
think it's just as you scale up, it becomes more and more logistically challenging. So we do have an experiment or a study at the moment that's using 3-meter tanks, which would be maintained at ambient and +4 degrees for 10 years. But I think for these sewage ones, it's because they're flocks, and we didn't want to filter all those out, because then again you lose diversity. But we know that these microcosms are relatively low diversity compared to the original inocular. But the fact that we see actually consistency across these different methods sort of reassures me, anyway, to a certain extent, that the diversity of resistance within that population is still sufficient to act as a reasonable model. But I mean, I think if you go back to the fact that all of ecotox testing in general, we want more and more environmental realism, but that's offset against reproducibility and and just how practical it is to run these experiments at scale.
Yeah. Have you ever tried to use different volumes and see whether you get
more
reproducibility when you're increasing the volume?
No, we just tend to use more replicates.
Okay, so now we'll continue with the next speaker, Chiara Borsetto from Warwick.
Hello everyone, and first of all, thanks to the organizer for giving me the opportunity to present some of the work that we've been doing with our collaborators in India, looking at the effect of pharmaceutical manufacturing waste on the environmental resistance and potential implication for human health. I won't go much into detail, but we are all very much aware about the problem of environmental pharmaceutical pollution that has implication for human health and antimicrobial resistance, and it has been shown that we have pharmaceutical all around the globe in rivers, And in particular for this project, we were mainly interested in the production of pharmaceuticals and how the waste might impact the environment. As we know that with waste being released in the environment, we can have a high concentration of pharmaceutical in rivers, for instance. India is one of the largest pharmaceutical manufacturers, and actually in January 2020, a new bill has been released to limit the discharges of antimicrobials in the environment from production plants. From the project, we were mainly interested in looking at the risk linked to AMR from pharmaceutical manufacturing waste being released in the environment, and how we do this is by, with our collaborator, we selected 2 sites in India, in Himachal Pradesh, in the north of India. Badi, which is our pharmaceutical manufacturing hub, where we know we have several different manufacturers. And we compare it to Kangra, which is a more residential area where we know there is no manufacturing actually happening. And we do that by sampling the environment and looking for active pharmaceutical ingredient and the effect that these might have on the environmental resistome. But we're also trying to look at the effect that the environmental microbiome might have on the human microbiome. And we do that by sampling also fecal samples from a donor at these 2 locations and looking at the resistome. We're very much aware that the human gut microbiome is exposed to pharmaceutical ingredients through different routes. So through the social science team, we are also looking at consumption data as well as other routes of exposure. But it's a very much complicated picture that we're trying to capture. As part of this talk, I will mainly present the data that we collected on the environmental samples. So in terms of sampling campaign, in 2019, we did a very small pilot study, so prior to the introduction of the bill. And then in 2021, when the project officially started, we had a few exploratory campaigns. And then from 2022 onwards, we have done a 3 sampling campaign where we selected 10 key sites in Badi and 5 in Kangra. And we collected mainly river water, wastewater from industrial areas, as well as some sewage and drinking water. And in terms of analysis, we've done resistant and mobile analysis using the high-throughput qPCR approach using the resistant map service, as well as metagenomic analysis. And we've also monitored API, looking at what we have in our samples through an untargeted approach for API identification, and we've also quantified heavy metals. So what have we observed so far? In terms of API pollution, what we could see was that Kangra was relatively cleaner comparing to Badi, as we could only identify mainly plasticizer. But in Kangra, in Badi, we had a much different picture, as we could identify several API, and in particular, these API were mainly identified from waste coming from industrial area, as we were expecting. But it's important to notice that actually from 2019 to 2022, for instance, we have noticed significant changes in the API that we could identify. And in particular, in 2019, we could identify several antibiotics, even at high concentration. But the only antibiotic that we could identify later on was rifampicin, and then all the other were compound with different activities, such as, for instance, antihistamine. So what we could see here was that we had potentially some changes in the manufacturing in Badi happening that we were not aware of, but also potentially some changes in the waste disposal. And so the team, the social science team at Bristol is actually now looking into these to try to have data in terms of production, so where and when, and also monitoring what has happened in this site. So in terms of resistome, what we could see was that there was a clear separation between Badi and Kangra. And this separation was mainly driven by a set of samples that were linked to wastewater from the industrial area, and also samples that were geographically in proximity of the waste stream from the industrial site. So the same site that we have actually seen presence of several API. And in these samples, we could also see a much higher abundance of ARGs and MGEs linked to anthropogenic pollution, but also some clinically relevant ARGs. So we are also very much aware that fecal pollution could potentially be a significant driver for ARGs in the environment. So we wanted to test whether there was any significant difference between the pollution that we see embodied to Kangra. So we use bacteroidetes as a sort of a marker for fecal contamination. And when we tested it, we could see that actually there was no significant difference between the two sites. Nevertheless, we can still see that we have a much higher prevalence of ARGs in body comparing to Kangra. So then the next question was to try to have a look at what is actually present in the gut microbiome of people living in these areas. So that's why we tested also fecal samples. And through metagenomic, what we could see was that there is a higher prevalence of ARGs in the fecal samples in body comparing to Kangra. And now we are also looking at MAGs to try to better identify the genetic context of these genes and see how these might be shared across the two environments and what is the relationship between them. So as I said, we have collected several different data that we are now trying to combine to better understand the effect of pharmaceutical waste released in the environment and then potentially the effect on human health. So my colleague Emma Travis tried to identify the major contributor to AMR at this site, and using this simple model, because it's just something that we just literally started, we could see that there was a site, a few sites seems to be driving AMR, and these are linked to industrial wastewater and the river in close proximity to the industrial site, but also sewage samples are also driving AMR as well as the mobilome. So our next step is actually trying to introduce the additional data that we are still trying to collect on API and heavy metals to try to better understand the relationship. So in conclusion, what we could see was that there was a significant reduction in the detectable antimicrobial residues in the environmental samples compared to the 2019 sampling campaign. We still see some API coming from waste stream samples from the industrial area that are also affecting river water in close proximity to this area. And these changes might be due to manufacturing changes as well as waste disposal changes. We could also see that there was the fecal pollution might be actually driving some of the environmental resistome, but still the pharmaceutical waste seems to have an effect in selecting more prevalently ARGs and MGs. And we are, as I said, we are trying now to integrate also the consumption data and other data that the social science team has been collected on, at the two locations to try to better confirm the actual route of exposure and also the effect that this might have on human health. And with this, I would like to thank, hopefully, all the people that have participated in this project. It has been quite challenging, but hopefully now we are starting to see some interesting results. And thank you all for your attention.
Dian Hua from University of Queensland, Australia. So I saw in the pharmaceutical manufacturing site, you do have an anti-tumor agent, right?
Yes.
So did you see any correlation between the anti-tumor drugs and the AMR occurrence? Yeah.
So that's actually something that we are exploring now. So I don't actually have an answer yet. Some of the data are actually still in the process. So hopefully maybe at the next EDAR I might be able to present the full story. But thanks, very interesting.
Any other questions? Oh. Thomas?
Yeah, next to I'm Thomas Berendonk, Theodresden Hydrobiology. You might have said that and I might have missed it, but you showed that in the where the production plant was, you had a higher prevalence in the fecal samples. So if you would look at the treatment plant effluent, is that then also higher?
So we haven't actually monitored the wastewater treatment plant, but we've monitored downstream of the wastewater treatment plant, and we don't actually see the same level.
Ah.
And the next speaker is Oriane Della-Negra from UNEA, France.
Okay, so hello everyone. So I'm Oriane Della-Negra, currently in a postdoc position at UNEA in France. And I'm going to talk about the evolution of antibiotic resistance in soils in presence of increased concentrations of sulfamethoxazole and clarithromycin. Yes. Okay, so I won't be long about it, but in the context of water scarcity, we will use more and more treated wastewater for irrigation. And the problem here is that treated wastewater may contain different antibiotics at different concentrations. And through irrigation, they can be transferred to soil. And depending on their persistence, on the irrigation condition, and the treatment for wastewater, they could accumulate and contaminate the soil with concentrations that can be found at microgram or 0.1 milligram per kilogram of dry soil. And the residual presence of these antibiotics could apply selection pressure and disseminate antimicrobial resistance in the environment. So the purpose of the study here was to investigate the effect of antibiotic nature and concentration on the evolution of the soil endogenous microbial communities, some key antibiotic-resistant genes, mobile genetic elements, and the fate of the antibiotics in soils. And to do this, we studied two antibiotics, sulfamethoxazole and clarithromycin, that are usually found in treated wastewater, and that harbor different physicochemical properties and that are associated with different antibiotic resistance mechanisms. So to do this, we used the soil microcosm approach. So basically some flasks filled with agricultural soil and irrigated in one shot at the beginning of the experiment with treated wastewater, spiked with clarithromycin or sulfamethoxazole, to reach final concentrations in soils of 0, 10, 100, 500, and 1,000 micrograms per kilogram of dry soil. So we applied the sacrificial mode, performed biological triplicates and several controls, and we measured, after the irrigation one shot, the temporal dynamic of soil bacterial communities, some key antibiotic-resistant genes, mobile genetic elements, and degradation genes, and the fate of the antibiotics and their transformation products over time. So I will just present here the main results obtained in this study in the next slides. So first, about the antibiotic biotransformation. So for sulfamethoxazole, I represented here the dynamic of the normalized concentration of sulfamethoxazole over time for the 4 tested concentrations. And what we could see here is that after 1.5 months of incubation, we had complete transformation of sulfamethoxazole for the 4 tested concentration. So we could apply a first-order kinetic model, determine the K kinetic constant and the DT50, the time needed to transform half of the antibiotic. And what we could see here is that we had a key concentration between 100 and 500 micrograms per kilogram of dry soil, from which we had a stimulation of the degradation with a decrease of the DT50, with a decrease of the time needed to degrade the sulfamethoxazole that could be associated or not with sulfamethoxazole inactivation. For clarithromycin, a completely different behavior, a strong decrease at the beginning of the experiment, and then a steady state for the 3 incubation months. So incomplete degradation that could be attributed here with clarithromycin absorption into soils that could limit its bioavailability and so its biodegradation by soil microorganisms. So about the transformation products, so it's a representation of the main transformation products that were identified in the sulfamethoxazole experiment. So basically, one color, one transformation product. So it corresponding here to the normalize the peak area, and the gradient color, the color gradient, sorry, corresponding to the sulfamethoxazole concentration gradient initially introduced into different microcosms. So first, at the beginning of the incubation, we observed the desamination product of sulfamethoxazole, then the hydroxylation products. So 2 transformation products with antimicrobial activity were found in literature to be higher than the parent molecule, that are usually associated with abiotic transformations or co-metabolisms. And there was also a 3rd transformation product that were identified here, and that corresponded to the N-glucuronidation of sulfamethoxazole, whose isomer was found to be quite persistent in soils, up to 83 days in other experiments. And that is usually a transformation that can facilitate some xenobiotic transport via efflux pump. About clarithromycin, even if we didn't have complete degradation, we observed 2 main transformation products. So in red, the transformation corresponding to the well-known phosphorylation of clarithromycin, responsible for the loss of its antimicrobial properties. And another one that I didn't represent it here because the peak area was too high, but which corresponding to the abiotic hydrolysis of clarithromycin that were also observed in controls. So then we searched for the potential effect on the soil bacterial communities with statistical analysis and linear effect size discriminant analysis. And we've identified some biomarkers associated with inhibition and selection effects. So inhibition of several taxa, such as Flavisalubacter on the left, Noviherbaspirillum, whose relative abundance decreased with increasing sulfamethoxazole concentration, but we also identified several selection effects, so increasing relative abundance with increasing sulfamethoxazole concentration, and more specifically of Hydrogenophaga and some unclassified Burkholderia and Rhodocyclaceae, sorry. And the main effect, inhibition and selection effect, were observed within the first incubation week. After this week, after 14 incubation days, we did not identify any major inhibition and selection phenomena in our samples. For clarithromycin, no significant taxa were identified, no significant inhibition effect or selection effects. So it could be due to the limited the absorption of clarithromycin in soils, which would limit its bioavailability, and maybe which would also limit its antimicrobial properties, or it could also be attributed to global tolerance of the soil microbial community to clarithromycin. Then we monitored the dynamic of some antibiotic-resistant genes. So I just show you for sulfamethoxazole the dynamic of INTL1 and SUL1. And what we could observe is that we had a transitory phase between 1 and 7 incubation days. As you can see, we had a quite nice correlation between the initially introduced sulfamethoxazole concentration and the relative, but also I didn't show it here, but we had the same results for the total abundance of these genes. So with 3 levels, 0, 10 microgram per kilogram of dry soil, then 100, 500, and 1000 microgram per kilogram of dry soil. For INTL1, we had a global increase of the abundance of this gene, even in control without sulfamethoxazole, that could be associated with treated wastewater input containing other antibiotics that could also apply a selection pressure. And finally, at the end of the experiment, even if we did not identified any selected taxa, we could see here that we had still 2 levels of abundance of INTL1, but also SUL1 genes that would suggest here some resistance transfer within the soil bacterial population. Okay, just to show you here, for example, I draw the temporal dynamic of some unclassified Rhodocyclaceae here. So you can see that the selection effect is observed between 1 to 7 incubation days. And I won't detail it, but we also performed some correlation linear correlation matrix on the 0-7 days time period, and we could see here that we had a quite strong correlation between the previously identified selected taxa and INTL1 and SUL1 gene abundance. So it would suggest, once again, his hypothesis, but that this selected bacteria could be the carriers of the sulfonamide-resistant genes in this study. And here I would like to attract your attention that we had a co-occurrence between the apparition of some transformation products of sulfamethoxazole and the increase of the relative abundance of INTL1 and SUL1 gene. So we could wonder if these transformation products could be responsible or could participate to the maintenance or the increase of this the abundance of these resistant genes in our soil microcosm here. And okay, and finally for clarithromycin, so last slide for this result, we've also observed some phenomena among ERMB and MEFA genes. So with ERMB, we had, after treated wastewater input, a global increase of these genes. And we could observe an effect of the addition of clarithromycin. So as you may see, we had a slight increase between the control without clarithromycin and the other concentrations, but no nice correlation as we could observe before. Because as I said it before, it could be due to the global tolerance of the microbial community or the limited bioavailability, which would limit also the antimicrobial property of the clarithromycin. And for the other gene, for the microlide efflux gene, we could observe some U-shape effect with increasing relative abundance, and once again, increasing total abundance of these genes for concentrations of 10, 100 microgram per kilogram of dry soil, and then decrease or no effect for higher concentration, 500, 1,000 microgram per kilogram of dry soil. So we could select some low abundance for these concentrations that were inhibited for higher concentrations. So finally, here in this study, we identified temporal windows associated with bacterial selection, inhibition, but also transformation product formation and some antibiotic-resistant gene dynamics. So bacterial and chemical indicators of sulfamethoxazole and clarithromycin contamination. So now, this is a microcosm approach, not real conditions. We could wonder what would be the effect of repeated and long-term irrigation in real conditions, but also what about the other soils, because of course, depending on the physicochemical properties of the soil type, we would have different results. And we also raised the question of the role that transformation products could have in the maintenance and the increase of antibiotic resistance. And finally, we have seen that with different antibiotics, we have different concentration thresholds, different behavior, and we would like to study more in depth the role between molecule adsorption, the bioavailability of the molecule, and the different degradation and resistance mechanisms here. So thank you for your attention.
Hello, Yue Huang from the University of Hong Kong. I'm working on biodegradation of sulfonamide, so my question is, did you do some investigation on the sulfonamide degradation gene? Is there any correlation with the sulfonamide resistant genes in your study?
Yeah, so I also monitored some sulfonamide degradation genes, so SAD-A, with the sulfonamide monooxygenase, but also the NAT gene, NR-lamine acetyltransferase. But we did not, for NAT gene, we quantified it, but we didn't have any effect of sulfamethoxazole concentration. And for SAD-A gene, it was below the quantification or the detection limit. So, and we did not identify any transformation products related to these transformations.
So I wonder if you know what happened with the sulfa functional group, in a long term. Is it known what happened with that sulfa group, and whether it still selects for, let's say, Enterobacteriaceae? Select for resistance for Enterobacteriaceae. In other words, whether the products of the sulfamethoxazole degradation still keep selecting for the sul gene in pathogenic bacteria.
Yes. So I made. Yeah. I'm not sure that I know everything about the literature, but we know that the two transformation products I showed at the beginning had a higher antimicrobial properties compared to sulfamethoxazole, but I don't know about the potential selection effect and the effect of antibiotic resistant genes, and this would be something to investigate, yes, in the future.
Thank you.
Thomas Heberer from the Federal Office of Consumer Protection and Safety from Germany.
Yeah, thank you so much for having me. And it's the second last talk today in the last session, so it's not that easy to provide anything new to a conference which was so excellent, giving so comprehensive information. So I try my best, because I've got one aspect which has, in my opinion, not yet been addressed at all. And I want to show you something about regulation, so what we already did in Europe. And just to address the issue of antimicrobial resistance, which is, of course, a very interesting and very urgent issue that we are addressing in Europe. So let's start with this overview. And I think we talked about humans, environment, livestock animals. But did we talk about companion animals yet? Not really, didn't we? So in our opinion, companion animals are also an issue for environment and also for the transference of antibiotic resistance to humans. And just to give you an impression, I'm living close to a lake, and a very beautiful lake, used for bathing of humans. Not allowed to be used for bathing of animals, of dogs, but nobody cares. And of course, it's an ecosystem where we are closely linked, and if you think about companion animals, it's a way different situation. So if you think about livestock animals, of course, farmers and veterinarians have a close contact to these animals, and they are in danger of getting resistance from these livestock animals. But companion animals have an even closer contact to humans. And what are we talking about? I was just checking the numbers again. In Germany, we have 15 million cats. We have 10 million dogs. And I know some microbiologists, and they should know better. I asked them, what are you doing with your cats and dogs? Are you taking them to your bed? And they said, yeah, of course. They like to do this. Weird. Wouldn't do this anyway. So there's a close contact, and I think nobody really cares. So we care about things that sound very dangerous. Sometimes we do things that are dangerous, and we don't even care. So anyways, you have seen these slides many times before, and of course there is an interaction between all these different areas. And so in Europe, we do something already for a longer time. So just to say this in advance, of course, Europe has first world problems. And of course, we have first world solutions which might not be applicable to all the other parts of the world. So these are just the measures that have been taken in Europe, and I just want to show you what we already did. And the first thing that we did on European scale, I know in Sweden we are pretty much advanced. So we had a ban of the use of, of antibiotics for growth promotion already in January 1st, 26. What else did we do? In 2012, we had a communication by the CVMP. It's a committee which is aligned to the European Medicines Agency, which is now located in Amsterdam. And the final conclusion was that we had a ban of third and fourth generation cephalosporins for poultry already in 2012. So we speeded up, and as you know, we have a new regulation in the EU. It's called 2019-06, and it has to be applied since January 28 of 2022. This doesn't mean that all the measures that have been said right there are already in place. So I can show you something that we already did and something that's still on the way. So, but there were two main objectives. First objective was reduce administrative burden. Probably not for the NCAs like our NCA, so we have even more administrative burden in the meantime. But it should be a reduction of administrative burden. Don't ask me for it. Second thing was main issue to strengthen the EU action to fight AMR. So we have a European strategy on AMR, we have national strategies on AMR. In the meantime, we have a EMA, HMA, Heads of Medicines Agencies strategy, and we also have one issue right there. One main issue is AMR. So what are we doing right now? So first of all, if we think we are just responsible for the authorization of veterinary drugs, so also, of course, of antimicrobials, and we have in Article 8 data to be submitted with the application. Why do I show you this? Because right here we have now implemented, as you can see it, and 2A, documentation on the direct and indirect risks to public or animal health or to the environment of use of the antimicrobial veterinary medicinal product in animals. This is new. So this is something that has to be filled, of course, with life in the future. What else do we have? We have Article 37, highly struggled from both sides. For one side it was not enough, for the other side it was way too much. So what are we talking about? So we talking about decisions refusing marketing authorizations. The Commission shall, by means of implementing acts, designate antimicrobials or groups of antimicrobials reserved for treatment of certain infections in humans. So that's a human reserve list that has been established. And it has been established in July 19th, 2022. And that's the list. So the one side complained, wow, it doesn't really make a change for food-producing animals. No, but it makes a real change for companion animals, because it's not allowed in Europe to use any longer carbon P names for animals. Not at all. Pretty new. Ban of carbon P names, for instance. All the others that are listed right here. What else do we have? Article 57 says collection of data on antimicrobial medicinal products used in animals. Right now it's mandatory to collect data both on volume of sales and volumes of use. So we already had a sales acquisition before, but now we also have use data. So we collected sales data until 2011 in Germany, and I can show you the result. We have a decrease in the sales of antibiotics in Germany by 68%. So from 1,700 tons down to 500 tons. Real success. We are not the only country, of course. All the countries in Europe reduced the amounts of sales of antibiotics significantly. For instance, Spain also by 70%, others by 50%, whatever. So it's a real success, but this is right now mandatory. So formerly we had ESWAG, and now it's called ASU, Antimicrobial Sales and Use Data Collection by Article 57. So what else do we have? We have Article 106.7, many articles. Use of antimicrobials in medicinal products. So this is all about off-label use. So it is said, well, it is possible, and this is a proposal by CVMP again, that compounds should not be used off-label under certain conditions. None has been identified to be never used off-label, but we have some compounds and compound combinations that can only be used under certain conditions, either for companion animals, for food-producing animals, and for aquatic animals. So we have susceptibility testing as a mandatory test to be done before using such combinations, for instance. Or sometimes with poultry, they're not allowed to be used in this case, first case, or not to be used for food-producing aquaculture. These are just two examples out of many other examples that have been initiated. So what else do we do at all? So we have a reinforcement of the ban of the use of antimicrobials for growth promotion. So this was just the old 2006 issue reinforced again. We have a ban on preventive use in groups of animals. So that's prophylaxis. We have restrictions on the use of antimicrobials in a metaphylactic way. We have the obligation of member states to collect sales and use data, as mentioned. We have the possibility to reserve certain antimicrobials in humans, as shown by this one of the previous slides in the list. And we have, what I've been mentioned, this issue of off-label use, which has also been restricted somehow. So usually in Europe, all the medicines have to be used according to the SPC, to the summary of product. Sorry, got it now. So it's a leaflet finally. So what's written in the leaflet, it has to be fulfilled, and you cannot derive from leaflet, with a few exceptions. Product characteristics. So this is what is now restricted very heavily, and it's not the end. I think we go any further, and if you see, we also implemented something on the environment. So thank you so much for your attention. This is just a brief overview on what was happening and what's still on the way. And we tried to keep all the input from this conference also to our agency. And well, I'm also part of the European network, and I'm in charge of the antimicrobial resistance in this network. And I thank you so much for all your presentations, for all the ideas that I got right now, bringing it back to Europe. And thank you so much for your attention.
Hi, I'm Aimee from Canadian Food Inspection Agency. I guess I have a question regarding this. Do you guys have data about the sales of non-human antibiotics such as ionophores? And also, do you guys have any data showing or any restriction policies about the use of ionophores as gross promotion and disease prevention?
So when we're talking about, so what we did in the past was just to collect sales data. So these all accumulated. So we had, of course, it was not mandatory. Now it's mandatory, and we are collecting use data right now since January 1st, 2023. Not very long. So we don't have yet published any data on use. And we have a tiered approach. So we started first with food-producing animals, and this will be extended finally also to cats and dogs. It's also different between the different member states. So we have not yet any data on cats, dogs, companion animals for use, unfortunately, but we are expecting it to come soon. So it probably answers your question. I wasn't really sure.
I mean, like specifically for ionophores, like monensin and lactalacid.
Yeah, we have data, of course. The problem is that we have to accumulate these data for protection reasons. So we can't tell you exactly which compound has been used at what amount because it touches some industrial properties finally. So this is a real problem. So we have to accumulate these data. Yeah. So, but you can see some data compared to different groups on our website. It's also available in English, so you can just take a look at the website and you'll find probably what you like to see, but maybe not because sometimes it has to be accumulated.
Okay, thank you very much.
You're welcome.
Yeah, so the reduction in the use of antibiotics for animals is quite impressive. Has anyone looked at the effect on that in terms of resistance levels in farm animals, for example?
Yes, we try to. So it's a very good question. Thank you so much for this question. So first of all, to mention, the reduction is really impressive. The problem is we started too early. So the Commission decided that we have to reduce by 50% later. So I think it was in 2018. Sometimes I miscompare. 2018, yes. My staff members were nodding. So by 2018, we have to reduce by 50%. Compared to 2018, we are now with 25%. But as far as your question is concerned, so we have a monitoring on animal pathogens going on since 2022. And so we have some data over time, and we see, of course, changes. But the problem is that these changes that we see have a certain lag time between 3 to 5 years, and that's a real issue. So you don't see it at once, so you see it later. And we see that there are some differences, but it's not that striking. So I couldn't answer that's really striking. And of course, what we see is a shift in compounds that are used. So you have several compounds, especially those used at higher doses. Of course, they have been reduced more than those that are administered at lower doses, which finally comes to an issue that is also addressed here at the conference. I think sometimes it's hard to put it just on figures by tons or anything else, because we should probably better be talking about daily doses, and this is not what we are doing. So we're talking about tons, we're talking about compounds that have way different doses that are administered. So we have, of course, for those who are administered at lower doses, not these significant reduction than we have for others, which is, yeah, just understandable from a certain point of view. So we have other measures in place to probably have some restrictions for the use of those antibiotics which are seen as more problematic, more interesting for human use. And something that I always have to explain to my students is we have now no antibiotics that are not used with humans in veterinary medicine. So no classes of antibiotics. Classes of antibiotics are not used with humans. So if you probably prohibit the use of antibiotics administered to humans for animal use, you have nothing left. Not an option. Also in view of potential zoonoses.
So now we'll go with our last speaker, Andreas Hanner from Roche.
I'm working as an environmental risk assessor at the Roche, and I'm also co-chair of the Pharmaceuticals in the Environment Task Force of the 3 European Pharma Associations and member of the manufacturing working group of the AMR Industry Alliance. And in this context, I would like to present you the antibiotic manufacturing standard, an industry approach for responsible effluent management in the context of antibacterial resistance. But first, as Thomas also talked about the EU legislation, I would like to first talk about the general pharmaceutical legislation package for human pharmaceuticals in Europe. It's a package which is currently completely changing, so renewed. And secondly, I would go to this antibiotic manufacturing standard of the Industry Alliance, then a little bit talk about the WHO guidance and sum up in the end. This general pharmaceutical legislation is now, it's a draft. We have a proposal of the European Commission from 2023, April, and we have a Parliament position of April 24, and we don't know exactly what's happening in the Council. That will be interesting to see, and when we have the opinion of the Council, then we will see in these trialogue discussions what will come out. But let's start with the blue one on the left side. I don't read everything, but we have firstly, for the first time for human pharmaceuticals, the definition for antimicrobials. These are antibacterials, antivirals and antifungals, and the Parliament added also the antiprotozoals. So I heard in the last days a lot about antibacterials. 2 days ago we have had 2 excellent speeches, talks about antifungals. I haven't heard much about or nothing about antivirals and antiprotozoals. And I mention that because for all these antimicrobials, we have to include resistance selection in the environmental risk assessment. That's really the first time it's appearing. I did a risk assessment voluntary 2 years ago. The EMA rejected that because it's not a legal requirement. But now it will come when it's in force for the entire, for the life cycle, for the entire manufacturing supply chain, inside and outside the union, the use and disposal. And it should take into account existing international standards that have established PNECs for the resistance selection. And another point which was added by the Parliament is, so the environmental risk assessment for antibacterials will come suddenly in place when the law is in force. But they say also that for the other antimicrobials, you will come up within 12 months with a guidance for these other antimicrobials. And you see, we do not know much about antivirals. Antifungals, I can imagine that we are quite faster, but for the other 2 groups, it's a lot of science we still need. And to sum up here at this point, we need a harmonized guidance for assessing risks for antibiotics, and we need the science for the other antimicrobials, so that in the end we have a guidance that you really can do this environmental risk assessment. So let's now move to the AMR Industry Alliance. It's one of the largest private sector coalitions established to provide sustainable solutions to AMR. It's based on 4 pillars. It's on access for everybody, for all over the world, for high-class antibiotics, and for diagnostics. It's about appropriate use in combination with vaccines and diagnostics. It's about manufacturing, effluent management, and it's about research and science. So really to push R&D activities. Now I would mostly like to focus on the manufacturing pillar, where I'm also working in. Manufacturing, it's one source of antibiotics in the environment. We have other sources. So at the global level, it's mostly excretion by patient, which is the most common reason for the contaminants in the environment, or inappropriate disposal just down the drain. But locally, we have seen, and it was mentioned in the literature a lot of time, locally you can have a big problem when you have wastewater which is not adequately managed. So we have seen that in several contributions this week. That's why the AMR Industry Alliance came up firstly in the beginning with a manufacturing framework, which described responsible effluent management, described also the adherence to PNEC values. It was a scheme which was established with a self-control. We self-reported our progress in our fields. We regularly, every 1 or 2 years, we said, what about our sites? What about our supplier sites? But it was a self-declaration. And that moved then now to this antibiotic manufacturing standard. You can download it under this QR code, manufacturing standard. And we implemented a third-party verification that these PNECs are met in the environment. The third party is actually it's BSI. In future, it will be other audit organizations which will do that as well. And now we are in the progress so that we are promoting this standard and this certification scheme, which shall be opened for all producers, not only members of the AMR Industry Alliance, all producers all over the world, which can seek now this certification. The standard, the scope is, in scope is the antibiotic manufacturing of drug substance, the APIs, and of drug product, the galenics. The general principles are the antibiotic API or drug product are made in accordance with robust environmental management systems, which are designed and consistently operated to minimize the loss of antibiotics into the environment. And the PNEC has to be met in the receiving water. The PNECs which we are applying in our standard are on the one hand the PNEC MSC values, which we heard, which were developed by Johan Pengsten-Palme and Joachim Larsen, 2016, and which were adapted by the AMR Industry Alliance and published on the Alliance website. It's a QR code there where you can download all these PNEC values which we have published. And the PNEC for the environment, they have been collated by industry. These are mostly GLP data which were assessed by the partners. So here you have an extract of this PNEC list from the internet. We published our approach in a paper, and we published also in a second paper default predicted no effect target concentrations in case we do not have any data. So that means we are working with a quite low value of 0.05 micrograms per liter, and then it's up to the companies to assess better data when it's needed for risk assessment purposes. About the certification, at the moment, we have 24 certified products and 5 more product certifications expected in the next time, and more than 70 products in the BSI, as a British Standard Institute pipeline. And the certification actually is issued to products made in 7 countries, and we have now 11 other countries which are awaiting this certification. So far in the 7 countries, you already have India included, so we have already Indian producers, and in these 11 countries, we already have Chinese producers which are waiting these certification steps. So we are engaging now, we are promoting that, so we would like that at least all the members really push that on their sides and mostly also on their supplier sides, so that in the end we can say that all our antibiotic supply chains are certified by a third party. Also, we consider it as an independent certification, which also leads to an increased transparency. The standard and certification scheme was acknowledged by many organizations as well, and it's really it gives us a push to promote that. I would like to show you now what would be the effect when we really apply this standard. So you have seen this scheme sometimes already this week. Joachim already showed it in his presentation on Monday. We just saw it also in this session. And when we apply our standard, so we are bringing down the concentration to 0.1 to 1 micrograms per liter, most of these substances. And when we see, so we have a median PNEC value of 0.25 micrograms per liter, we significantly bring down the concentration. And that's a worst case, because in most cases you are below the PNEC values, because in many jurisdictions you are not allowed to fill up to the PNEC. You have to apply the best available technique first, and you are not allowed just to fill up to the PNEC. So shortly to the WHO guidance, which was developed by the end of last year. So we have a mass balance approach, and we apply our PNECs in the receiving water. The basic scenario of the WHO guidance is also based on the mass balance, but it also applies the PNEC in the mixing zone, but at the fixed dilution rate of 10, whereas in the antibiotic manufacturing standard, we have the local scenario. On the right side, it's a stringent, very stringent scenario where you have the application of the PNEC end of pipe and only chemical analysis allowed. So we are highly we were not included to build up this WHO guidance, but we could send comments to them, and our view is that this antibiotic manufacturing standard is somehow similar to the basic scenario of the WHO guidance. There are some differences, of course. It could be something which could be in a compromise be solved, I think. But we think that the WHO stringent scenario would be too costly to really implement it as a only standard for antibiotic production, and it's up to an environmental risk assessment where we think that should be in place. So we have from the Scandinavian initiatives, we have the backup for the Nordic countries, Norway, Denmark, Finland, and Iceland, which implemented that in their procure system. This standard and the certification scheme, which will lead to the maximum amount of points, and this gives us a good backup for us for our work. So we are acknowledging that how critical it is to tackle AMR, and has been active in seeking robust means to drive this solution. We need a harmonized guidance for to assess the environmental risks for antibiotics for the full life cycle for German pharmaceutical legislation. We need a common guidance for this responsible manufacturing effluent management. We think that independent certification offers a robust means to provide stakeholders with accurate antibiotic manufacturing and show that it's everything controlled. But as I said in the beginning, we also need harmonized guidance for antimicrobials other than antibiotics. Here the science is missing. So thank you very much to my colleagues, Joan Elmoznino, which is sitting in the audience, of Pfizer, and Kirsty Reed of FBR, and Steve Brooks of the AMR Industry Alliance. Thank you very much.
And I had two, but maybe I'll choose one. Thanks a lot for presenting the initiative here, Andreas. I have a technical but still, I think, important question, and that is the mass balance approach for estimating discharges. That is the only thing that is required in the industry standard. So I get it really how you do it when you do formulation. You calculate how much API you put in, and you see how much you get out, and then unaccounted losses is what you assign to the water. But how do you go about when you have fermentation-based production? Because there's no API coming in. It's bacteria or fungi that creates the API amount, which is basically unknown. And then you get an amount that you get at the end. How do you calculate the losses when you don't really know what's coming in?
You have completely right there. You can only go to a theoretical concentration, because there you really cannot do the mass balance. Yeah. Because there you have to somehow go to the highly expected yield which you really can expect. Otherwise it's not to be possible. But that's really a weak point with the mass balance, I agree.
And if I may, a completely different question. So transparency came up here, and there's been a discussion on transparency in the pharma production sector for the last 15 years. What do you think it will take for industry to be publicly transparent about both production lines, so the suppliers, who they are, where production takes place, and emission levels? Because I think that would be wanted by many.
Ooh, that's a difficult question. And it's difficult for me to speak for the whole pharmaceutical industry, but I think, yeah, I could imagine that it's that sometime that really the names are published as in this new when it's possible in this New Zealand website. It's not always very accurate what's there, because it's only there what is reported, but at least it's quite good. But I think that will be difficult because, yeah, there are other jurisdictions, there are other companies. It's not the suppliers are not our companies somehow. So we are the clients of them, and I don't know. Very difficult.